Today we're going to talk about pathogens. Now, just a reminder, this mind map is already linked down below, so make sure to grab your copy and annotate as we go. Pathogens are part of our area of study 2 in Unit 3. Really what they are any disease-causing agents, and the ones that we focus on are infectious. This means, as I'm sure we're very aware with COVID going right now, that the infectious particles or these agents can be transferred through the environment. There's many ways that this can happen via touch, fluid, water sources and vectors, but we're not going to look into that too much in this video. Instead, what we're going to focus on are the 5 main groups of pathogens. Before we start though, it's really important that we go over 2 main definitions. Those 2 are disease and infection. Now I want you to think in your layman's terms, in your general life, you've probably used these interchangeably. So you've kind of swapped and used them as the same word. What I want us to really delineate or show the difference between is that a disease is a condition or change within an organism which impairs their functioning. So it means that no longer is the body or the cell going to work as it usually should. An infection, then, is the presence or multiplication of a disease-causing agent within an organism or a cell. And you can have this infectious particle or this disease-causing agent multiplying and existing within an organism without having any of the detrimental or negative impacts. And at that point, it's simply an infection. And so really, what I want us to see is disease are the negative impacts and infection is simply the presence of a pathogen. Okay, in biology, it's really good to section things off so you remember how they work. And if we look at the whole pathogens, there's an easy way to start off with it. First of all, we have virus and prions on the right, which are acellular, and bacteria, fungi, parasites, and protists on the left, which are cellular. Hopefully you know that cellular means that they are going to be living. Cells are the smallest unit of functioning life, and A, if anything starts with A, it means without, so non-living. This is good to remember how they function, but especially significant for our first group, the virus. This group is submicroscopic, so they're smaller than is visible by the naked eye, which means we can't see when someone coughs or sneezes or the little viral particles flying around. They also contain their own genetic material, and this can be RNA or DNA, but it's very specific to the type of virus that it is. The type that I've drawn on the left here, you can see that I've indicated that there's genetic material within this caspid, this protein layer, and then this outside layer has little markers or receptors which do connect to the host cell, and that's actually derived, this layer, from a previous host cell membrane. So as they've undergone exocytosis, they've taken some of that membrane with them. Because they are part of this non-living group, they actually require a host cell to replicate. They're not able to reproduce on their own, and they come in and they actually take over host cell machinery to replicate. This includes the ribosomes, for example, to take that genetic material, transcribe it, or if it's RNA already, to just translate it into the proteins that are required to make the future viral particles. It's important as well, particularly for Unit 4, to think about the fact that these are not treatable by antibiotics. We can split up biology words, usually read them backwards. So split it up, we've got biotics for bacteria and anti for against. Instead, what these particles need are antiviral. What can be seen, or what's drawn at the top here, is a bacteriophage. That's another way that you'll see viruses drawn. But again, we can split up the word and we see that the bacteria refers to the fact that these are usually found, or will always be, viruses that affect bacteria. Our next group are the prions. We don't know as much about this group, but what we do know is that they are submicroscopic as well. They're actually simply made up of small proteins. They cause degeneration of the nervous system, and they have no genetic material. Really what they are, it's just made up of small proteins with only tertiary structure, so it's only one polypeptide chain. They have alpha helices and beta pleated sheets. And they malfunction or they become pathogenic in that they have a misfolding to them. And I'll go through an example in a moment. But what's interesting about these is that they could be inherited, but they're also transferred through the consumption of infected tissue. So that's why as those of you who are from England or know anything about it, know a little bit maybe about mad cow disease and how we had to stop eating red meat or a lot of meat for a period of time, because they would be able to potentially transfer these prions, which could cause degeneration of our nervous system. Not great. I'm going to run us through an example that affects sheep, not humans, and that's called scraping. Now, if we remember prions, it's all about misfolded proteins. So for this scraping, there are two forms of the protein. There is PRPC, the prion form, normal, and PRPSC, the scrapy form, abnormal. The PRPC is usually found on the surface of neurons, so those cells that are making up our nervous system. They're important, or these cells in particular are important for memory, learning, and neuronal signaling. What happens is that when a PRPSC, so the scrapy form, encounters a regular form, it actually causes the regular protein to undergo a conformational change in the tertiary structure. So where there used to be more alpha helices, they then change and become beta pleated sheets. These malformed proteins then aggregate, they clump together on these neurons and mean that no longer do they function as they should. So really, again, it's that structure related to function, the fact that just this change in tertiary structure or secondary really is creating such a great difference in this protein's function. If we now move over to our parasites and protists, these are kind of clumped together, they're a bit irregular. They also don't get really tackled in the VCE curriculum or exam context that often. So what we just really need to know is, again, they're pathogenic, so they're going to cause a negative effect in an organism. They can be unicellular or multicellular. They can invade host cells or organs, tissues, more widespread. And we've got a few examples, but they're all going to be eukaryotic. Okay, examples include malaria, ticks, and amoeba. Really, as I said, it's a very large grouping, and so we can't make too many general statements about it. And so if they ever do ask about this, it's about pulling the information out of the question. So into our next group of cellular or living pathogens, we have our fungi. We typically think about mushrooms, but when we're talking about our pathogens, they're usually microscopic. They typically produce skin or surface infections in humans, and they are eukaryotic, again, but with chitin walls instead of our cellulose-based plant cell walls. And then, of course, animal cells don't have cell walls. They reproduce via spores, and they're treatable by fungicides. I would suggest not Googling images of these, but examples of fungi include tinea and ringworm. And so into our final group of pathogens. Out of the 5, this one, alongside the viruses, are the ones that you're going to see pop up most, particularly when we're doing responding to antigens and immunity. These bacteria are microscopic, so they're a little bit larger than viruses. They are prokaryotic. Hopefully you already know what that means in terms of the fact they have no membrane-bound organelles. They have a circular piece of DNA, and they're very small, and they are also unicellular. They can, however, be formed and found in colonies of a really large number of them, and they replicate quite rapidly. Really though, bacteria are typically harmless. The large majority exist in our body in a way that doesn't affect us. So we have infections, we have large numbers of them, but they don't create disease. It's really interesting to think about the fact that right now in your mouth, there are actually more bacteria than there are people on Earth, and that's by quite a significant amount. So bacteria are not inherently bad. However, when they do cause damage, they can directly destroy tissues and cells, but usually it's actually that they're harmful through the chemicals that they produce. And what we say is that these are called toxins. So through their normal metabolism, they release chemicals into the environment around them, their extracellular environment, and those toxins and those chemicals can damage tissues and cells. They, as you might already know about bacteria, reproduce through binary fission. They are treatable by antibiotics, so again, bacteria against, and they are uncommon in plants as the sap is too acidic. This is going to be significant when we look at our first line of defense in plants coming up soon, so just keep that one in mind. So there's our 5 main groups of pathogens. We've got our non-cellular viruses and prions, and our cellular bacteria, fungi, parasites, and protists. When we think about all of these, we're now going to move into area study 2, responding to antigens, and how our body actually fights infections of these particles, particularly when they start to cause those negative effects of disease. Thank you for watching. Don't forget to like and subscribe. Get that mind map from below, fill it in, stick it up on your wall, and definitely let us know which videos or which information you'd like us to make resources on.